Book Review Design Principles of Biological Circuits by Johns Wentworth, 5th of November, 2019. I remember seeing a talk by a synthetic biologist almost a decade ago. The biologist used a genetic algorithm to evolve an electronic circuit, something like this.
Image. A dense, machine-evolved electronic circuit diagram is shown in a rough monochrome schematic style, packed with transistors, resistors, capacitors, and interwoven signal paths. The arrangement appears highly irregular and difficult to follow, with components connected in seemingly unintuitive ways rather than the clean, modular structure typical of human-designed circuits. Labels such as Q1, Q18, R1, R14, and various voltage markers are scattered throughout the diagram, reinforcing the impression of a functional but opaque design whose purpose is not immediately apparent to a human reader.
He then printed out the evolved circuit, brought it to his colleague in the electrical engineering department, and asked the engineer to analyze the circuit and figure out what it did. "I refuse to analyze this circuit," the colleague replied, "because it was not designed to be understandable by humans." He has a point. That circuit is a big, opaque mess. This, the biologist argued, is the root problem of biology. Evolution builds things from random mutation, connecting things up without rhyme or reason into one giant spaghetti tower. We can take it apart and look at all the pieces. We can simulate the whole thing and see what happens, but there's no reason to expect any deeper understanding. Organisms did not evolve to be understandable by humans. I used to agree with this position. I used to argue that there was no reason to expect human intelligible structure inside biological organisms, or deep neural networks, or other systems not designed to be understandable. But over the next few years after that biologist's talk, I changed my mind, and one major reason for the change is Alon's book, An Introduction to Systems Biology, Design Principles of Biological Circuits. Alon's book is the ideal counter-argument to the idea that organisms are inherently human opaque. It directly demonstrates the human understandable structures which comprise real biological systems. Right from the first page of the introduction.
One can, in fact, formulate general laws that apply to biological networks. Because it has evolved to perform functions, biological circuitry is far from random or haphazard. Although evolution works by random tinkering, it converges again and again onto a defined set of circuit elements that obey general design principles. The goal of this book is to highlight some of the design principles of biological systems. The main message is that biological systems contain an inherent simplicity. Although cells evolved to function and did not evolve to be comprehensible, simplifying principles make biological design understandable to us.
It's hard to update one's gut-level instinct that biology is a giant mess of spaghetti without seeing the structure firsthand, so the goal of this post is to present just enough of the book to provide some intuition that, just maybe, biology really is human understandable. This review is prompted by the release of the book's second edition just this past August, and that's the edition I'll follow through. I will focus specifically on the parts I find most relevant to the central message. Biological systems are not opaque. I will omit the last 3 chapters entirely, since they have less of a gears-level focus and more of an evolutionary focus, although I will likely make an entire separate post on the last chapter, Evolution of Modularity. Chapters 1 to 4, Bacterial Transcription Networks and Motifs. E. coli has about 4,500 proteins, but most of those are chunked together into chemical pathways which work together to perform specific functions. Different pathways need to be expressed depending on the environment. For instance, E. coli won't express their lactose metabolizing machinery unless the environment contains lots of lactose and not much glucose, which they like better. In order to activate/deactivate certain genes depending on environmental conditions, bacteria use transcription factors, proteins sensitive to specific conditions which activate or repress transcription of genes. We can think of the transcription factor activity as the cell's internal model of its environment. For example, from Alon.
Many different situations are summarized by a particular transcription factor activity that signifies, "I am starving." Many other situations are summarized by a different transcription factor activity that signifies, "My DNA is damaged." These transcription factors regulate their target genes to mobilize the appropriate protein responses in each case.
The entire state of the transcription factors, the E. coli's whole model of its environment, has about 300 degrees of freedom. That's 300 transcription factors, each capturing different information and regulating about 4,500 protein genes. Transcription factors often regulate the transcription of other transcription factors. This allows information processing in the transcription factor network. For instance, if either of two different factors, XY, can block transcription of a third, Z, then that's effectively a logical NOR gate. Z levels will be high when neither X nor Y is high. In general, transcription factors can either repress or promote, though rarely both, and arbitrarily complicated logic is possible in principle, including feedback loops. Now we arrive at our first major piece of evidence that organisms aren't opaque spaghetti piles, bacterial transcription network motifs. Random mutations form random connections between transcription factors. Mutations can make any given transcription factor regulate any other very easily. But actual transcription networks do not look like random graphs. Here's a visualization from the book.
Image. The image compares two small network diagrams side by side. On the left, a real network is arranged in a branching, hierarchical structure with relatively orderly connections and clustered subgroups. On the right, a randomized network with the same number of nodes and arrows appears much more tangled and irregular, with connections crisscrossing unpredictably between nodes. Both diagrams include 10 nodes and 14 arrows, but the randomized version has fewer self-reinforcing loops, emphasizing the visual contrast between structured and random connectivity.
A few differences are immediately visible. Real networks have much more auto-regulation, transcription factors activating/repressing their own transcription, than random networks. Other than self-loops, aka auto-regulation, real networks contain almost no feedback loops, at least in bacteria, though such loops are quite common in random networks. Real networks are mostly tree-shaped. Most nodes have at most a single parent. These patterns can be quantified and verified statistically via motifs or antimotifs. Connection patterns which occur much more frequently, or less frequently, in real transcription factor networks than in random networks. Alon uses an E. coli transcription network with 424 nodes and 519 connections to quantify motifs. Chapters 2 to 4 each look at a particular class of motifs in detail. Chapter 2 looks at auto-regulation. If the network were random, we'd expect about 1.2 plus or minus 1.1 auto-regulatory loops. The actual network has 40. Chapter 3 looks at 3-node motifs. There is one massively overrepresented motif, the feed-forward loop, see diagram below, with 42 instances in the real network and only 1.7 plus or minus 1.3 in a random network. Distinguishing activation from repression, there are 8 possible feed-forward loop types, and 2 of the 8 account for 80% of the feed-forward loops in the real network. Chapter 4 looks at larger motifs, though it omits the statistics. Fan-in and fan-out patterns, as well as fanned-out feed-forward loops, are analyzed. Alon analyzes the chemical dynamics of each pattern and discusses what each is useful for in a cell. For instance, auto-regulatory loops can fine-tune response time, and feed-forward loops can act as filters or pulse generators.
Image. The image shows two simple 3-node network diagrams placed side by side under the title, "The Two Overrepresented Feed-Forward Loop Motifs." Each diagram consists of nodes labeled X, Y, and Z, connected by directional arrows forming triangular patterns. The left diagram, labeled Coherent Type 1 Feed-Forward Loop, shows all connections aligned in a consistent direction of influence, while the right diagram, labeled Incoherent Type 1 Feed-Forward Loop, includes one opposing interaction, visually indicated by a different arrow ending. The layouts emphasize two closely related but functionally distinct patterns of connectivity.
Chapters 5 to 6, Feedback and Motifs in Other Biological Networks. Chapter 5 opens with developmental transcription networks, the transcription networks which lay out the body plan and differentiate between cell types in multicellular organisms. These are somewhat different from the bacterial transcription networks discussed in the earlier chapters. Most of the overrepresented motifs in bacteria are also overrepresented in developmental networks. But there are also new overrepresented motifs, in particular positive autoregulation and two-node positive feedback. Both of these positive feedback patterns are useful mainly for inducing bistability, i.e., multiple stable steady states. A bistable system with steady states A and B will stay in A if it starts in A, or stay in B if it starts in B, meaning that it can be used as a stable memory element. This is especially important to developmental systems, where cells need to decide what type of cell they will become in coordination with other cells, and then stick to it. We wouldn't want a proto-liver cell changing its mind and becoming a proto-kidney cell instead. After discussing positive feedback, Alon includes a brief discussion of motifs in other biological networks, including protein-protein interactions and neuronal networks. Perhaps surprisingly, especially for neuronal networks, these include many of the same overrepresented motifs as transcription factor networks, suggesting universal principles at work. Finally, Chapter 6 is devoted entirely to biological oscillators, e.g., circadian rhythms or cell cycle regulation or heartbeats. The relevant motifs involve negative feedback loops. The main surprise is that oscillations can sometimes be sustained even when it seems like they should die out over time. Thermodynamic noise in chemical concentrations can kick the system so that the oscillations continue indefinitely. At this point, the discussion of motifs in biological networks wraps up. Needless to say, plenty of references are given which quantify motifs in various biological organisms and network types. Chapters 7 to 8, Robust Recognition and Signal Passing. There's quite a bit of hidden purpose in biological systems. Seemingly wasteful side reactions or seemingly arbitrary reaction systems turn out to be functionally critical. Chapters 7 to 8 show that robustness is one such hidden purpose. Biological systems are buffeted by thermodynamic noise, and their functions need to be robust to that noise. Once we know to look for it, robustness shows up all over, and many seemingly arbitrary designs don't look so random anymore. Chapter 7 mainly discusses kinetic proofreading, a system used by both ribosomes, RNA reading machinery, and the immune system to reduce error rates. At first glance, kinetic proofreading just looks like a wasteful side reaction. The ribosome/immune cell binds its target molecule, then performs an energy-consuming side reaction, and just waits around a while before it can move on to the next step. And if the target unbinds at any time, then it has to start all over again. Yet this is exactly what's needed to reduce error rates. The key is that the correct target is always most energetically stable to bind, so it stays bound longer, on average, than incorrect targets. At equilibrium, maybe 1% of the bound targets are incorrect. The irreversible side reaction acts as a timer. It marks that some target is bound and starts time. If the target falls off, then the side reaction is undone and the whole process starts over. But the incorrect targets fall off much more quickly than the correct targets. So we end up with correct targets enriched. The fraction of incorrect targets drops well below its original level of 1%. Both the delay and the energy consumption are necessary in order for this to work. The delay to give the incorrect targets time to fall off, and the energy consumption to make the timer irreversible. Otherwise, everything just equilibrates back to 1% error. Alon offers an analogy in which a museum curator wants to separate the true Picasso lovers from the non-lovers. The Picasso room usually has about 10x more lovers than non-lovers, since the lovers spend much more time in the room, but the curator wants to do better. So, with a normal mix of people in there, he locks the incoming door and opens a one-way door out. Over the next few minutes, only a few of the Picasso lovers leave, but practically all the non-lovers leave. Picasso lovers end up with much more than the original 10x enrichment in the room. Again, we see both key pieces, irreversibility and a delay. It's also possible to stack such systems, performing multiple irreversible side reactions in sequence in order to further lower the error rate. Alon goes into much more depth and explains the actual reactions involved in more detail. Chapter 8 then dives into a different kind of robustness, robust signal passing. The goal here is to pass some signal from outside the cell to inside. The problem is, there's a lot of thermodynamic noise in the number of receptors. If there happen to be 20% more receptors than average, then a simple detection circuit would measure 20% stronger signal. This problem can be avoided, but it requires a specific and non-trivial system structure. In this case, the main trick is to have the receptor both activate and deactivate, i.e., phosphorylate and dephosphorylate, the internal signal molecule, with rates depending on whether the receptor is bound. At first glance, this might seem wasteful. What's the point of a receptor which undoes its own effort? But for robustness, it's critical, because the receptor both activates and deactivates the internal signal. Its concentration cancels out in the equilibrium expression. That means that the number of receptors won't impact the equilibrium activity level of the signal molecule, only how fast it reaches equilibrium. The trick can also be extended to provide robustness to the background level of the signal molecule itself. Alon provides more detail. As you might expect, this type of structure is a common pattern in biological signal receptor circuits. For our purposes, the main takeaway from these two chapters is that just because the system looks wasteful/arbitrary does not mean it is. Once we know what to look for, it becomes clear that the structure of biological systems is not nearly so arbitrary as it looks. Chapters 9 to 11. Exact adaptation, fold change, and related topics. When we move from an indoor room into full sunlight, our eyes quickly adjust to the brightness. A bacteria swimming around in search of food can detect chemical gradients among background concentrations varying by 3 orders of magnitude. Beta cells in the pancreas regulate glucose usage, bringing the long-term blood glucose concentration back to 5 millimolar, even when we shift to eating or exercising more. In general, a wide variety of biological sensing systems need to be able to detect changes and then return to a stable baseline across a wide range of background intensity levels. Alon discusses 3 problems in this vein, each with its own chapter. Exact adaptation. The output signal of a system always returns to the same baseline when the input stops changing, even if the input settles at a new level. Fold change. The system responds to percentage changes across several decibels of background intensity, extracellular versions of the above problems in which control is decentralized. Main takeaway: fairly specific designs are needed to achieve robust behavior. Exact adaptation. The main tool used for exact adaptation will be immediately familiar to engineers who've seen some linear control theory. Integral feedback control. There are 3 key pieces. Some internal state variable M, e.g. concentration/activation of some molecule type or count of some cell type, used to track error over time. an internal signal, X, an external signal, and a receptor, which increases production/activation of the internal signal whenever it senses the external signal. The error, tracked by the internal state M, is the difference between the internal signal's concentration X and its long-term steady-state concentration X star. The internal state increases or decreases in direct proportion to that difference, so that over time, the M is proportional to the integral of X star minus X dt. Then, M itself represses production, or activation of the internal signal X. The upshot: if the external signal increases, then at first the internal signal X also increases, as the external receptor increases production or activation of X. But this pushes X above its long-term steady-state X star, so M gradually increases, repressing X. The longer and further X is above its steady state, the more M increases, and the more X is repressed. Eventually, M reaches a level which balances the new average level of the external signal, and X returns to the baseline. Alon then discusses robustness of this mechanism compared to other possible mechanisms. Turns out, this kind of feedback mechanism is robust to changes in the background level of M, X, etc. Steady-state levels shift, but the qualitative behavior of exact adaptation remains. Other, simpler mechanisms do not exhibit such robustness. Fold change detection. Fold change detection is a pretty common theme in biological sensory systems, from eyes to bacterial chemical receptors. Weber's law is the general statement. Sensory systems usually respond to changes on a log scale. There's two important pieces here. Respond to changes means exact adaptation. The system returns to a neutral steady-state value in the long run when nothing is changing. Log scale means it's percent changes which matter, and the system can work across several orders of magnitude of external signal. Alon gives an interesting example. Apparently, if you use a screen and an eye tracker to cancel out a person's rapid eye movements, their whole field of vision turns to gray and they can't see anything. That's responding to changes. On the other hand, if we step into bright light, background intensity can easily jump by an order of magnitude. Yet a 10% contrast looks the same in low light or bright light. That's operating on a log scale. Again, there's some pretty specific criteria for systems to exhibit fold change detection. Few systems have consistent, useful behavior over multiple orders of magnitude of input values. Alon gives two particular circuits, as well as a general criterion. Extracellular/decentralized adaptation. Alon moves on to the example of blood glucose regulation. Blood glucose needs to be kept at a pretty steady 5 millimolar level long term. Too low will starve the brain, and too high will poison the brain. The body uses an integral feedback mechanism to achieve robust, exact adaptation of glucose levels, with the count of pancreatic beta cells serving as the state variable. When glucose is too low, the cells slowly die off. And when glucose is too high, the cells slowly proliferate. The main new player is insulin. Beta cells do not themselves produce or consume much glucose. Rather, they produce insulin, which we can think of as an inverse price signal for glucose. When insulin levels are low, so the price of glucose is high, many cells throughout the body cut back on their glucose consumption. The beta cells serve as market makers. They adjust the insulin/price level until the glucose market clears, meaning that there is no long-term increase or decrease in blood glucose. A very similar system exists for many other metabolite/hormone pairs. For instance, calcium and parathyroid uses a nearly identical system. Integral feedback mechanism using cell count as a state variable with a hormone serving as price signal to provide decentralized feedback control throughout the body. Alon also spends a fair bit of time on one particular issue with this setup. Mutant cells, which mismeasure the glucose concentration, could proliferate and take over the tissue. One defense against this problem is for the beta cells to die when they measure very high glucose levels, instead of proliferating very quickly. This handles must mutations, but it also means that sufficiently high glucose levels can trigger an unstable feedback loop. Beta cells die, which reduces insulin, which means higher glucose price and less glucose usage throughout the body, which pushes glucose levels even higher. That's type 2 diabetes. Chapter 12. Morphological Patterning. The last chapter we'll cover here is on morphological patterning, the use of chemical reactions and diffusion to lay out the body plans of multicellular organisms. The basic scenario involves one group of cells, A, producing some signal molecule, which diffuses into a neighboring group of cells, B. The neighbors then differentiate themselves based on how strong the signal is. Those nearby A will see high signal, so they adopt one fate, while those farther away see lower signal, so they adopt another fate, with some cutoff in between. This immediately runs into a problem. If A produces too much or too little of the signal molecule, then the cutoff will be too far to one side or the other. E.g., the organism could end up with a tiny rib and big space between ribs, or a big rib and a tiny space between. It's not robust. Once again, the right design can mitigate the problem. Apparently, one group ran a brute force search over parameter space, looking for biologically plausible systems which produced robust patterning. Only a few tiny corners of the parameter space worked, and those tiny corners all used a qualitatively similar mechanism. Alon explains the mechanism in some depth, but I'm gonna punt on it, much as I enjoy non-linear PDEs, and this one is even analytically tractable. I'm not going to inflict them on readers here. Once again, though it may seem that evolution can solve problems a million different ways and it's hopeless to look for structure, it actually turns out that only a few specific designs work, and those are understandable by humans. Takeaway, let's return to the Alon quote from the introduction.
Because it has evolved to perform functions, biological circuitry is far from random or haphazard. Although evolution works by random tinkering, it converges again and again onto a defined set of circuit elements that obey general design principles. The goal of this book is to highlight some of the design principles of biological systems. The main message is that biological systems contain an inherent simplicity. Although cells evolved to function and did not evolve to be comprehensible, simplifying principles make biological design understandable to us.
We've now seen both general evidence and specific examples of this. In terms of general evidence, we've seen that biological regulatory networks do not look statistically random. Rather, a handful of patterns, motifs, repeat often, lending the system a lot of consistent structure. Even though the system was not designed to be understandable, there's still a lot of recognizable internal structure. In terms of specific examples, we've seen that only a small subset of possible designs can achieve certain biological goals: robust recognition of molecules, robust signal passing, robust exact adaptation and distributed exact adaptation, fold change detection, robust morphological patterning. The designs which achieve robustness are exactly the designs used by real biological systems. Even though the system was not designed to be understandable, the simple fact that it works robustly forces the use of a handful of understandable structures. A final word. When we do not understand something, it does not look like there is anything to be understood at all. It just looks like random noise. Just because it looks like noise does not mean there is no hidden structure.
Image. The classic psychological image known as the Dalmatian dog illusion.